Today, we're honored to welcome Dr. Bill Anish to the show today. He's a world-renowned expert in longevity. I'm very excited to have him on the show today. He has over 50 patents in terms of longevity space. He has a lot of solutions that many people haven't really heard of or spoken about. So I'm very excited to have this topic and have him on the show. How you doing, man?
I'm doing okay, and I hope you're doing well too. I appreciate you letting me have an opportunity to speak during this podcast.
Absolutely. I'm very intrigued to hear your story and how you got into the longevity space. Did you have your own health journey? Like, how did it all start?
uh, well, actually, my health journey is no different than anybody else's. A lot of times you hear about people promoting some new way of doing things to cure a certain disease, and then the first thing they start doing is talking about how they actually suffered from that disease, and that's why they got so focused on trying to deal with that disease. I, I've I don't have that kind of history. I've just been obsessed since I was a little kid. I mean, I guess the best way to start off is saying that I was fascinated with science and medicine even when I was a child in elementary school. Okay, my teachers used to send me home with notes on my pinned to my shirt, and my parents would read it and it would say, "Your son is obsessed with science and medicine. You should nurture this." And then, so one day, my parents had bought me this really nice, for a 10-year-old, this was something unusual, but this like a 4-foot long, 8-inch diameter reflector telescope so that I could sit in the front lawn and look at all the moons of Jupiter and rings of Saturn and things like that. But one day, while I was doing that, my father came out to the front lawn and said to me, "Bill, when you grow up, you should become a doctor and find a cure for aging." And he said, "I don't know why nobody's done this before." And he said, he explained to me that this was something he's been really interested in his entire life. And so I took the baton essentially from him and made it my goal in life. And it's like, because I was like really fascinated with the whole subject of why do we age? It seems like nonsense for us to develop all this knowledge and experience and stuff like that and then have it all thrown away. One of my favorite colleagues, Aubrey de Grey, one time he and I were sitting down having some drinks. He drinks beer, I drink Diet Coke. But he said to me, "Every time a person dies, we lose as much information as is found in the Library of Congress in Washington, D.C." And that stuck with me. And I've been really thinking this is horrible. And then when we see all these people that can never be replaced, my favorite example is Betty White. I think one of the worst things that ever happened to this planet was the passing of Betty White. I mean, she was such a great entertainer. And no, there's never going to be another Betty White. I mean, and then there's also these geniuses and stuff like that that have really done some great things in the past, and they passed away. And shoot, and I don't think anybody's following in their footsteps to really carry the baton for them. I mean, it's like because everybody's so focused on promoting themselves, they don't want to promote somebody else's goals. I've never been like that. I would promote anybody's goals if I thought that they were really useful. But a long answer to a short question, but I've been. Let me just finish with try to speed it up. I went to, when I went to high school and college, all I could think about was, what do I need to learn in order to eventually find a cure for aging? And I tried to study all the causes of aging and stuff like that. And I started learning that none of them made sense because not all the twos and twos added up. I was taking courses in logic and statistical theory and just common sense and things like that, just trying to make sense out of this whole thing. And I studied evolution galore because I want to find out, why did we evolve this dumb thing called aging? Why have we never evolved a way not to age? So that's been my obsession in anti-aging clubs and stuff like that, especially in college, where some of the people that were sitting in these meetings with me won Nobel Prizes later on. I mean, I was working with a pretty elite group of scientists, and The but, you know, I don't know if I'm getting off subject, but we did come up with the conclusion that in terms of how we age, there has to be something inside of our cells that's like ride tickets at an amusement park. Every time a cell divides, you lose a ticket, and when you run out of tickets, you die of old age. Because everything we learned about biochemistry and all the biochemical pathways and things like that, and the mechanisms that cells use to function, we couldn't figure out how could a cell keep track of time. And the only way was something, every time a cell divides, it loses something. Okay? And that's what happened. And there's only a certain number of these things that can lose. That was the only kind of mechanism. And I'm going to stop there because I'm now going to start skipping to other questions you're probably going to be asking. But yeah, that's been me my whole life.
Thank you, man. I appreciate that. It's an awesome story. It sounds like your parents were kind of influential in your decisions in terms of longevity and cultivating that. Do I have that right?
Oh, absolutely. My father, after he saw that I really took the baton from him, he started going with me to anti-aging conferences. He was a television producer, most known for game shows like You Don't Say and It Takes Two, Liars Club, things like that. He was the #1 daytime television show producer
Wow.
for 7 straight years one time back then. But he's passed away in 2015 from Alzheimer's. So ever since then, I've been kind of working alone. But before then, I mean, he and I were like teammates in this whole endeavor to try to cure aging. He encouraged me and helped me as every way he could. Yeah, no question.
Wow.
My mother was interested too, and she passed away in 2020 just from general aging. But she listened to everything I ever had to say about how to extend lifespan and health span as long as possible.
That's awesome, man. Had that support. Like it's really kind of amplifies the mission, right? And now you're that for other people, just kind of leading the way. So thank you for sharing that. So in terms of longevity, can you define what that actually is for people?
Well, longevity is just a measure of how long we live and how healthy we live during all that time. Okay, none of my colleagues in the field of anti-aging are interested in making old people live long but not be healthy. Okay, everything's about maintaining your health span as long as possible. But that's what longevity is all about. The question is, why is our longevity limited? And how is it? What limits it? And that's what all of us are trying to work on. And I think we're making some serious progress. Now, I'm just jumping short. I think me and a bunch of us know exactly what needs to be done. The only obstacle is funding. It's a tremendous amount of funding that's needed to develop a cure for aging, but we know what to do. And all of us are working together. There's no such thing as competition by the really top scientists in the field of anti-aging because we're all interested in having our own aging cured, and we're not going to stand in the way of somebody else curing their own aging.
That's a good point. We're all working together for the same mission. And I heard you mention health span. Can you define the difference between lifespan and health span?
Well, yeah, it's like if you were to watch the movie Zardoz, which might be before your time, there was all about how aging had been cured. Nobody was aging. Everybody was living forever. But there was this place where the really, really oldest people were confined to beds with tubes inside themselves, but they were still living, and it was almost like they were praying to die. Okay, well, we don't want that. We want people to feel like, look, feel, and behave like they're 25 when they're 500 years old. That sounds crazy. Okay, I admit that, but it's like there's no question about the fact that we are obsessed with actually making that happen. And my motto is cure aging or die trying. And by my definition of cure aging is to extend my lifespan and health span as long as possible. So is a lot of other people that are I consider my colleagues.
100%. And there are certain markers of aging, right? Telomeres being probably one of the top ones. You want to define that?
Yeah, well, telomeres are one of the biomarkers of aging. I would say that there's probably 100 different biomarkers of aging. When you go into your doctor's office and he measures your aging, he'll do everything from hand grip strength to, vision and IQ and things like that. But the top ones lately have been telomere length, DNA methylation, and IgG glycosylation. IgGs are antibodies. Okay, so those and they get glycosylated, and the glycosylation patterns change with age, and they can be measured. So those three biomarkers are the top ones. There's also epigenetics, which is a little more difficult to measure, but we have a technique called we do what's called total RNA sequencing here to measure epigenetics. Epigenetics is just gene expression. Gene expression. So you have I forget how many genes a human has suddenly. I think it's 25,000 different genes, and they change in their expression. Genes are next to every gene is like a dimmer switch, turns genes on and off, and that changes with age, and that's what gives us our aging characteristics. And so measuring that is something that we do here, but it's not ready for everybody, for personalized medicine, for people to be testing it, because it's just real expensive. But telomere length, IgG glycosylation, and DNA methylation, which is an epigenetic mechanism. People call it epigenetics, but it's actually an epigenetic mechanism. But I would say that the toughest one to measure is actually telomere length, even though I consider telomeres to be the main regulator of aging. And I think that telomere length affects DNA methylation and IgG glycosylation. So measuring IgG glycosylation and DNA methylation are actually indirect ways of measuring telomere length. Because I haven't really gotten into telomeres yet in this podcast, but telomeres, I think, are the main clock of aging, and that's why I have diverted my attention to focus mostly on that, even though my whole life I've been interested in all possible causes of aging. I just decided a long time ago that aging will never be cured no matter what we do, unless one of those things we do is solve the telomere shortening problem. And I know we haven't gotten into that yet, but I will when you get ready for that.
Definitely. Thanks for sharing that, man. Would you consider sirtuins one of the top ones as well?
Oh yeah, well, sirtuins are a major biomarker of aging. They're not the easiest thing to measure, but they do. That's part of epigenetics. Okay, so so doing the total RNA sequencing, one of the things we do measure is sirtuin expression. Okay, expression meaning the turning on and off of genes. We've done sufficient studies showing that sirtuin expression can go up and down in exact correlation with telomere length being going short, long, short, long. So we believe that telomeres do play a role, are a major role in regulating how much sirtuin is produced in the body.
Got it. And when it comes to telomerase, that's an enzyme that can add length onto the telomeres. Is that right?
Yes. And that actually is one of the major tools that we use to study telomere biology. And so we've known that telomeres shorten with age for, so we haven't talked about telomeres, but we have to do that before we talk about telomeres. Okay, so telomeres are found at the very tips of your chromosomes. They're actually part of the chromosome. They're a DNA sequence that's TTAGGG, where T's and A's and G's are bases. They're like beads on a string. DNA is made up of 4 different beads called T, A, C, and G. And a typical chromosome is about 100 million bases in length. And it's the sequence of these bases that tell us what our eye color is, our hair color, how smart we are, things like that. Okay, so, and everybody's different. Okay, so everybody has a different sequence in their DNA. And then there's, like as I mentioned, 25,000 different genes along these beads of strings. And the beads are divided into 23 different beads because we have 23 different chromosomes. But so at the very tips, so think of that bead as like a shoelace. And at the very tips of your shoelaces, you have aglets, the caps on your shoelaces that protect your shoelaces from damage. And when they wear away, your shoelace falls apart. Well, telomeres are the same thing. They're at the very tips of our chromosomes, and when they get shorter, our DNA starts falling apart, essentially. But it's a lot more complicated than that. That simple analogy is a good first step in trying to understand telomere biology. But it turns out the telomere not only protects the chromosomes, but it also turns genes on and off. It controls these dimmer switches in the same way that enhancer sequences. So some of the audience in your podcast might know what enhancer sequences are, but they're sequences found within our chromosomes that can regulate a gene that's like 100,000 bases away by the DNA folding over. So let me get my hand. The DNA folds over, comes in contact with the dimmer switch on a very particular gene, and adjust that switch. Okay, telomeres can do the same thing. And so, but the big question. Okay, so I first learned about telomere shortening in the early 1990s, and when I heard the person who was speaking about it at a conference that my father was also at, I was stunned because this was the first time that I ever heard about anything that could be those ride tickets at an amusement park that we'd all decided years before that had to play some type of role in the aging process. So I went up to the guy, his name was Dr. Calvin Harley, and I went up to him before he could get him off the podium. I was so excited about this presentation that I asked him, has anybody figured out how to re-lengthen the telomeres, essentially adding ride tickets back? And he said, no, we've been working on it for years. We've had colleagues, collaborators working with us. None of them have been able to figure it out yet. And I just flat out said, let me come and work with you, and I'll have it figured out in 3 months. And because of my history, I've had a lot of success in biotech. I discovered like human growth hormone, tissue plasminogen activator, erythropoietin, beta-seron, and I've only listed like about a fraction of the number of things that I have played a role in inventing and co-inventor in. And so he ended up, I was also National Inventor of the Year for cancer research. So he ended up essentially hiring me on the spot. And then 3 months and 17 days later, I discovered this enzyme. My team discovered this enzyme called human telomerase. He was working at a company called Geron Corporation, and he hired me and then put 3/4 of the company reporting to me just to get this done, get this research done. And we discovered this enzyme called human telomerase. And then it took me 17 days longer than I said, and I blamed him for it, for him and other senior people in the company for distracting me too much. But that's just a joke. We did discover this enzyme, and the way we discovered it, okay, I mean, if you think about it, if every time a cell divides, the telomeres get a little shorter, and remember, telomeres are part of the DNA, then our children will be born with shorter telomeres than we have, because their reproduction, their making the sperm and the eggs, requires cell division. So they would be made with cells that had shorter telomeres if telomeres were really shortening in our reproductive cells. And then their children would be born with shorter telomeres than they have. And we would have been extinct as a species 300,000 years ago. So there had to be something going on in our reproductive cells to prevent telomeres from shortening. So that was the focus. We ended up discovering that our reproductive cells produce an enzyme called human telomerase. And when we put that gene, the gene for human telomerase, into other cells, skin cells for instance, we found that we could reverse aging in every way imaginable in human skin cells grown in a petri dish. That's not so exciting. But then I ended up leaving the company for reasons I'll come to later, but it had to do with the company's focus change. But some people decided to continue. And I left to start my company, which is CR Sciences, to focus on aging. Dr. Walter Funk, who was one of the people I was working with and actually shared some awards with, he ended up growing skin on the back of a mouse and then treating that skin with telomerase to lengthen telomeres, and found out that skin got younger in every way imaginable by every biomarker that existed at the day. And then Dr. Ron DePinto, who was at Harvard and later became the head of MD Anderson, he created a strain of mice. Okay, so mice don't age by telomere shortening. In fact, most animals age by different mechanisms. Okay, the only animals that age by telomere shortening that we've found so far are humans, non-human primates, dogs, cats, horses, sheep, pig, and deer. Practically everything else ages by a different mechanism, including all rodents. But he engineered rodents, and specifically mice in this case, so that they would age by telomere shortening. And he found that they got old in every way that you would imagine, just like in humans. And then he activated telomerase to lengthen telomeres, and these mice became younger in every way imaginable, including their memory coming back. Okay, for those that had suffered from dementia, couldn't remember how to go through a maze when they got old, they could remember. Okay, that's making diseases like Alzheimer's and stuff like that a key target for telomere lengthening. We believe that now that lengthening telomeres in people who are suffering from Alzheimer's could actually bring their memories back, which is saying that Alzheimer's isn't a loss of memory, it's just a loss of access to the memory. The memory is still there, we just lost the access. So that's one of the goals of my research, is to try to start seeing if we can cure Alzheimer's. So we discovered that enzyme telomerase, and since I had a strong cancer background, one of the things that I did was I developed inhibitors to telomerase enzyme, because cancers, in order for cancers to become big enough to kill you, they have to solve the aging process, okay, the aging problem. Okay, so they do that by chromosomal rearrangements and mutations that eventually turn on the telomerase gene. Since our reproductive cells produce telomerase, it says that all of the cells of our body have telomerase. The genes just shut off. And that's a whole long story that we should take an hour to explain too, why that happened. But our cancers end up to get, if you're over 30, or let's say over 50, in order for a cancer to get larger than a small pimple, it has to figure out a way to solve the aging process. Otherwise, it would die of old age before it killed you. So it turns on the telomerase gene by mutations and chromosomal rearrangements, and your cells start producing, your cancers start producing telomerase, and that's why the cancer gets big enough to kill you. So where was I going with that? I lost track of where I was going. What were we just talking about beforehand? Why did I bring up that subject of cancer?
You're talking about how the cancer becomes big enough to kill you?
Yeah, but there was some reason why I went into cancer. But it was, I hope you edit this confusion out. I've been having that problem my whole life. Okay, I get off onto tangents, then I forget where I was. Let me just think for a second. What was I just talking about beforehand? Okay, so telomerase. Oh yes. Okay, so I was talking about why I left Geron Corporation, which is where I was working. Okay, so when I was so because of my interest in cancer, I had developed inhibitors of telomerase to see what effect they would have on killing cancer cells. And so I created inhibitors, and it killed every cancer cell I tested it on, and had no effect on non-cancer cells. So Geron Corporation, the investors in Geron Corporation, decided there was a quicker return on investment by curing cancer than curing aging. And since I had already had a lot of background in cancer and a lot of research, and already developed a lot of drugs that have been through clinical trials and stuff like that, I didn't like that. Plus I felt like cancer is an age-related disease, and so the real cure for cancer is curing aging. So and so did the founder of the company and a few other employees. And when Geron Corporation told us to stop working on aging, I left and I started Sierra Sciences to continue that research. And I did it with Geron's blessing. Okay, and then a few people like Dr. Walter Funk remained at Geron Corporation and did like the skin on the back of the mouse experiments. So, but I made it really clear that I thought inhibiting telomerase would actually never really work, because by inhibiting telomerase in cancer cells, you're only going to make the telomeres get really short, which is only going to increase the mutation rates in chromosome rearrangements. And so whatever you treat the cancers with, they're going to mutate to some form that's going to allow the cancer to come back. And that's exactly what happened. Okay, so the main thing that I invented was something that I called GRN 163, and that ended up later becoming called Rytelo, R-Y-T-E-L-O, which just got approved in clinic by the FDA just a few months ago, maybe now a year ago. But it took 30 years to get that approved. Okay, and that's because, as I said, the cancers were always coming back, and so it was really hard to get it approved. And they got it approved simply on the fact that it does add some life to people. So here at Sierra Sciences, we're also working on trying to cure cancer, but we're doing it by poisoning cells that produce telomerase, not inhibiting telomerase itself. Okay, that way the telomeres remain long, the mutation rates don't skyrocket, and the cancers won't come back. That's a project that is limited only because of insufficient funding. But that's something that we're working on. So that's pretty much how I got to where I'm at right now. I've been running Sierra Sciences for a while, searching for ways of causing our cells to produce telomerase, even though the gene is shut off in all of our cells. And I said it would take an hour to explain why we have shut off the telomerase gene, but this is where my evolution background comes from, and I'll just say it in a few words. By eliminating the longer lived, you increase the chances of the offspring interbreeding as opposed to the parents rebreeding. And as a result, by letting the offspring interbreed, you increase diversity within our species, which increases our species' ability to have some members survive rapidly changing environments. So it turned out any species that evolved a way to eliminate the longer lived after they had raised their young had a better chance of surviving. And so species that didn't figure out a way to eliminate the longer lived have mostly died off. So most species today have ways to eliminate the longer lived, and there's a few that don't. That includes lobsters and humpback whales and tortoises. They have no detectable aging process, but probably in 100,000 years they will. Okay, some of it takes time, but that's pretty much why we have our telomerase gene shut off, and that's the same reason why dogs, cats, horses, sheep, pig, and deer also have the telomerase gene shut off, so that, well, I don't want to say so that, because that makes evolution sound like a force, not a result. The members of the species that did shut off the telomerase gene overtook the regular population. So the species that developed ways of eliminating the longer lived were more likely to survive.
100%, man. That's a lot of information there. It's really incredible.
And somebody's going to have to listen to that and go figure it all out, or they can listen to YouTube videos where I explain that in great detail in a user-friendly way. So it's like not I don't go into strong evolutionary formulas and things like that, but there's YouTube videos that I have that people can find on what aging is, how we age, why we age, and how not to age. But sorry, go ahead.
Awesome. Yeah, good tip there. I mean, that is the question you want to ask, right? You asked the question, has anyone found out how to add length back onto telomeres? And then you went and found out. That's awesome.
No, we have. The problem is that the tools that we use is called gene therapy, okay? And it's just not safe enough for human use. But it allowed us to do experiments with mice and human cells in a petri dish. But gene therapy involves delivering a new gene to the cells, okay? And that's not the safest thing to do. I mean, just the delivery mechanism of delivering any gene to a cell can kill a lot of the cells, and probably a lot of the mice, when Dr. Ron DePinto did these experiments, probably ended up not surviving the delivery. Okay, so we're focused on trying to find like natural products and things like that that can get inside of our cells and turn on the gene that's already there. Yeah, but so we can lengthen telomeres. We just can't do it in a safe enough way to make it available for the general public yet.
Very good. You also used an excellent analogy in terms of like the shoelaces and the aglets. That's like a perfect picture of what a telomere actually is to paint for people. And you went to cancer a little bit, and correct me if I'm wrong, but essentially that's a cell mutation, right? After, like, it's kind of a natural thing after the Hayflick limit, essentially replicating a certain amount of times, and then you kind of get this cancer cell. But it's kind of a natural process that our body can deal with if it's not like overrun. Do I have that right?
Yeah. Okay. So cancer has the problem of the Hayflick limit. Hayflick limit means Leonard Hayflick in late 1950s, and he published it in 1961, showed that human cells can't divide forever, like a bacteria or a yeast that can just keep dividing. Human cells can only divide a certain number of times, and then all of a sudden they stop dividing. So if you look at a growth curve, the growth curve goes up and then all of a sudden levels off. It's called the Hayflick limit. Most people didn't believe it at first. I sure believed it when I first learned about it in the early 1970s. But so the cancers have to deal with that Hayflick limit too. And so when they reach that Hayflick limit, the cancer would stop growing and never hurt you. But because of the skyrocketing mutation rates from telomere shortening, the cancers mutate to figure out a way to induce the production of telomerase, to lengthen the telomeres and overcome the Hayflick limit. And that's what our reproductive cells do too. They don't reach a Hayflick limit. When you take reproductive cells and divide them in a petri dish, they never reach a Hayflick limit, like our skin cells and other cells of our body do. Yeah. So, but let me point out one of the main reasons why we get cancer. One main reason we get cancer when we're longer lived is because of telomere shortening. But another reason is because our immune systems go into a phase called immune senescence, and that's also a result of telomere shortening. So our immune system quits functioning because telomeres get too short to allow it to function correctly. And that allows, and our best defense against cancer is our immune system. So we get cancer 7 times a day on average. Our immune system wipes it out. It doesn't work very well after you become longer lived or old enough to have immune senescence.
Interesting. And did I hear you correctly when you said we haven't really found a way to add telomerase back on via certain lifestyle factors, or have we?
Yeah, we have. Okay, so we have. We just haven't. Okay, so this is another analogy. Telomere lengthening and shortening is like a tug-of-war. So in our reproductive cells, think of a tug-of-war. We have people pulling to shorten our telomeres. We also have people pulling to lengthen our telomeres. And so in our reproductive cell, every time a cell divides, the shorteners pull to shorten. But then right after it divides, the lengtheners, which is telomerase, pull to re-lengthen it. So it's a back and forth. The tug-of-war is a tie in our reproductive cells. In all of the other cells of our body, our telomeres, we only have shorteners. So every time a cell divides, it gets shorter and shorter. And so the tug-of-war, the people pulling to shorten our telomeres have no competition with anybody. So what we have done is we have discovered nutraceuticals that will add people to the lengthening side of the tug-of-war, so that now there is some lengthening. We just haven't won that tug-of-war yet. Okay, and this is something that a lot of companies will say they have won the tug-of-war, and it's not exactly true. And it's not. I shouldn't say that it's not true at all. But we have discovered nutraceuticals, plant extracts that, when added to human cells, will lengthen. Okay, but shortening still wins the tug-of-war. But now we're losing that tug-of-war at a slower pace. So we are extending our lifespan and our health span by just having a few people on the lengthening side. What my ultimate goal is to tie and win that tug-of-war. And so we are doing our research to continue searching for things that are more potent, especially plant extracts, because they don't require FDA approval, plus they're natural. We're continuing to search. We've searched over 20,000 different plant extracts so far, but there's 3 million out there to test. The problem is this screening or testing costs us $2 million a month. Okay, and so it's hard to get that kind of funding, so we can't go at the pace we'd like to do this research. If we could get that $200 million a month, I think we would have something potent enough to win the tug-of-war in 3 years at the latest, 1 year at the earliest. 1 to 3 years from now, we would have something that could win that tug-of-war. And if we got really lucky, it would be sooner than 1 year from now. Then that would be available for the public right away. Now, but getting back, because that's my ultimate goal, and so I sometimes forget about the fact that we have things that slow down the rate of shortening and win the tug-of-war. And so we kind of, those ones we put on the shelf, and we use them to learn about how to maybe make the extract or purify things from the extract to make it more potent. But fortunately, companies like Touchstone Essentials come to us and say, hey, we are very excited about the work you're doing, and we think it's very important that you have something that could slow down the aging process, even though you're not reversing aging yet. We would like to put that on the market, make it available to people. And I'm okay with that. I was fine with that. We licensed it to Touchstone Essentials. I'm just not a marketer. I'm not a product developer. I'm just a researcher. So, excuse me for not having found somebody on my own to go and do that. Touchstone Essentials came to me asking me if they could license our things. So we license our 5 most potent, what are called telomerase inducers. They're the ones that induce the cells to start producing people to pull on the lengthening side of the tug-of-war. So we sold them we licensed them our 5 tops, and they put those together along with some antioxidants and anti-inflammatories that reduce the number of people on the shortening side of the tug-of-war. I forgot to mention, you can reduce the number of people on the shortening side of the tug-of-war with things like antioxidants and anti-inflammatories, but not completely. There's still this thing called the basal level rate of telomere shortening caused by what's called the end replication problem. So it can't get below that. But now, by adding people on the other side, we can actually reduce it even further. So slow down the aging process better than any antioxidant or anti-inflammatory could do. And we have the ability, with the continued research, to make something more and more potent. And I'm so sold on Touchstone Essentials, I've already told them that when we discover these other things, I want them to be the ones marketing it. But in the meantime, the Telovital is the name of the product that they put together. And as I said, I don't market, but I do explain the science. But they are marketing this, and I am very excited about it in hindsight. I should have thought of this before, but it is the best thing that anybody on the planet could do to slow down the rate of their aging. Even if they're doing something that causes accelerated aging, such as smoking, obesity, those things accelerate aging. There's genetics things that accelerate aging. Drinking also accelerates aging. But you still can compensate for that a bit by Telovital to lengthen the telomeres. And there are other products that do claim to lengthen telomeres, some that even claim to win the tug-of-war. But they don't realize that all they do is they measure telomere lengths. And as I said at the very beginning, telomere lengths are very difficult to measure, and they're also very confusing to interpret the results. Okay, but it turns out there's other ways to make it so that your blood telomere lengths are longer without actually lengthening the telomeres in your blood. And that's by taking cells away from the blood that have short telomeres, and any toxin will do that. Right? Cells with short telomeres are going to be more sensitive to toxins than cells with longer telomeres. So if you were treated with a toxin, that's going to kill off the cells that have short telomeres in your blood, and as a result, your average telomere length is going to be longer. Just as if you fired your dumbest employees in your company, the average IQ of your company is going to increase without anybody getting any smarter. I know the other thing is that anything that's immunogenic is also going to cause the length of your telomeres in your blood to be longer without lengthening, because what it's going to do is it's going to induce naive cells or memory cells in your bone marrow to enter the blood that already have longer telomeres. Even when you're 100 years old and you die of aging, there's cells in your bone marrow that still have very long telomeres. But those are dormant. They sit there waiting for reasons to fight an infection or something like that. You flood they invade your blood, and as a result, your telomeres in your blood get longer, just as if your company hired a bunch of geniuses. Okay, the average IQ of your company increases without anybody getting any smarter. So that's what's happening with a lot of these other companies that are claiming to lengthen telomeres. They aren't lengthening telomeres. They're actually saying that their product is probably toxic or immunogenic, and that's why telomeres are behaving longer. So I've been saying for 30 years, people, even Dr. Calvin Harley, who I worked with at Geron Corporation, he used to say that the only way to really show that you're lengthening telomeres in your blood is to show that you are producing the enzyme telomerase. So the product that you're taking, whether it's a supplement or a therapy or like treatment with different wavelengths of light or things like that, if you see that the telomeres are getting longer, you cannot say that they're being lengthened unless you can show that you are producing telomeres. So I go to all these companies that are the companies. I say, do you have any data showing that you are producing telomeres? And they say no. And then I say, why not? And they say, well, we haven't been able to do it. And I say, well, measuring telomerase is easy to do. I'm actually one of the inventors and co-inventors of every method of measuring telomerase expression and telomere length there is. So I know this, but they still can't do it. Or some say they've never tried. And then I'll say, why not? Okay, because that's what they really need to do to show that their product is actually lengthening telomeres. And so what I'm recommending, and there's a lot of YouTube videos where I'm speaking at medical conferences where I'm telling doctors, before you recommend that one of your patients take a product, ask the product manufacturer if they have data showing that their product is inducing the expression of telomerase. Because, I don't want to say if any of them do or not, because I don't want to badmouth any other companies publicly, but that's something to go and ask. If a company can't provide data showing that they are expressing telomeres, then don't believe that their product is lengthening telomeres. Believe that their product is probably toxic or immunogenic. So Telovital, of course, since I'm the one that provided the data, I have tons of data showing that the ingredients in their product are inducing the production of telomerase in normal cells of our body. But
Very
not
good,
enough
man.
to win the tug-of-war. Not enough to win the tug-of-war. And that's something I'm still working on.
Thanks for sharing that. It's incredible. I mean, having things that we can go to expand our health span and just feel better overall. It sounds like you want to have something that has been proven to increase telomerase, which is something you've done. So that's incredible.
Let me add something else that makes it even more interesting. We've shown, and so others have shown too, and it's been published, that when telomeres get really critically short, it's a lot easier for telomeres to lengthen them. Okay, so when I speak at conferences trying to explain this, I explain like a telomere isn't a linear string of beads like a shoelace. It's actually all wound up like a ball of yarn, and the tip of the telomere is buried inside that ball of yarn. So when the telomere is long, the tip of that telomere is very buried in that ball of yarn, and so it's hard for telomerase to get in there to lengthen it. But when the telomere gets really short, the ball of yarn essentially disappears, and now it's really easy for telomerase to get in and re-lengthen the telomeres until it gets long again, pulls up like a ball of yarn. So even though Telovital isn't producing enough telomerase to win the tug-of-war, it could be winning that tug-of-war when people have critically short telomeres. And a lot of people have critically short telomeres in some tissues of their body. In fact, anybody over like 70, 80 years old has critically short telomeres in all cells of their body. And it is very likely, we don't have data on this yet, but I'm very interested in following people taking this. There's very, very likely, based on published literature, that even Telovital will lengthen the critically short telomeres, thus providing some age reversal effects. And that's the only way to explain all the anecdotal data that we're seeing that suggests that. I gotta confess, I have another meeting in 7 minutes right from now, so somehow I lost track of time. What would you like to do? Would you like me to tell the other meeting that I'm going to be delayed, or?
No, I respect that, man. Thanks for letting me know. If you can go over briefly, like what are some lifestyle factors that people are engaging in that are shortening their telomerase, and some things they can do to increase it?
Well, smoking, obesity, and drinking are the top ones. Okay, and like drinking's an easy example to explain because it's killing liver cells, and we all know that. And then so people would have to watch my videos on how we age to understand this, but as a result, telomeres shorten faster in our liver cells because of the killing of the cells in our liver. And so we don't see any effects when we're young because we still have cells that can divide to replace the killed cells. But when we get to be 40, 50 years old, we lose the ability, because of short telomeres, to be able to divide and replace the killed liver cells. And so we get liver cirrhosis, which is accumulation of dead cells. But the same is true for like smoking to our lung cells. Obesity is straining a lot of different tissues in our body, especially our blood vessels. But then there's also genetic things like muscular dystrophy, causes rapid changes in our killing of our muscle cells. We don't observe muscular dystrophy when we're young. We wait until it doesn't start showing up until we're older because we've accelerated the rate of the telomere shortening in our muscle cells. But when you get to be so even young people, 50, 60, can possibly see some age reversal effects. The anecdotal data has been lots of people getting better vision and lots of people seeing hair color come back and hair coming back. Okay, that can only happen if the decreased vision or the loss of hair or the loss of hair color was a result of telomere shortening. So not everybody's going to see it, but there is a surprising number of people. I personally have seen my endurance get better. I'm an ultramarathon runner, so it's easy for me to measure my endurance abilities. But my vision has gotten better too. In fact, I sometimes find out I have to take my glasses off to read things. But even though I have bifocals, those not shown here. So things you can do to decrease the rate of telomere shortening, in addition to taking Telovital, is meditation, yoga, decreasing antioxidants, anti-inflammatories, even though both of those are in Telovital, and just a lot of different things. And a lot of YouTube videos, I go over a whole lot of that.
Awesome, man. Thank you so much. Dr. Andrews, this has been incredible. Thank you for sharing your story, your expertise in telomerase, and your discoveries and all that, man. I really enjoyed this interview. And if people want to get connected with you, where can they go?
Oh, I want to give out my email address. I think I've already given it to you. Do you already have it? Otherwise, I can give it to you now.
I'll see if I can get that linked up.
I really want to answer all the questions that people have. My email address is B as in boy, Andrews, at Sierra Sci, that's SIERRASCI, short for Sierra Sciences, .com. Okay, and so I'm really good at responding to emails when people ask me for advice, because I'm just obsessed with making the world happier and healthier. That's my only motivation. I'm not in it for making money, things like that. So people can count on me getting back to them.
Awesome. Thank you so much for coming on and sharing all your insights with us. It's been awesome.
All right. Well, thank you. All right.